Hey guys, Mr. Hyatt here. This is part 2 of the Chapter 8 AP Biology lecture. In part 1, we left off on this slide, and I was kind of rushing because I was up against the clock, and so I think I did a poor job of explaining this slide. So notice down here when we look in box C at our net delta G, which is our net change in our free energy. When we look at the non-catalyzed reaction, the delta G is plus 3.4. So that means that it's going to require an energy input to make this reaction happen. It's not going to be a spontaneous reaction. In box B, you can see that we've used ATP to activate that glutamic acid, and that takes energy. There's our plus 3.4 kilocalories per mole. But we also, through the process of splitting ATP into ADP, we make up some ground. So we end up with a net delta G that's negative, which is going to mean this reaction is going to happen spontaneously. So we've lowered our activation energy, we've pushed the reaction forward. Okay, so we need to be real careful, especially when we are answering free response questions relating to photosynthesis, about the word food. Okay, ATP is going to be a renewable energy source. It's got those unstable phosphate groups that those bonds are going to break and reform and break and reform, and it essentially works like a battery in our bodies. Food is different. Food is going to be long-term energy storage. They're going to be stable bonds. Food implies that you are consuming it. So especially when you're writing responses to questions about photosynthesis, be real careful about saying things like plants make their own food. Technically, plants don't make their own food. They make their own energy. Certainly, ATP is going to cycle. Like I said, we can break it apart, reform it, break it apart, reform it. When we have anabolic reactions, remember anabolic reactions are building things from smaller molecules. Energy is going to be released, and that's going to drive those anabolic reactions. So we're going to have to consume energy to build things. Then when we're breaking things apart, ATP is going to be reformed and recharged by the free energy from catabolic reactions when we're tearing things apart. Here's a picture that kind of shows that process. Energy from catabolism, when we break things down and their energy is released, that energy is going to reform ATP. It's going to do this arrow. Then we're going to add water and hydrolyze and dephosphorylate, and we're going to release free energy to do endergonic reactions, to do anabolic reactions, and we're going to reform our ADP and our inorganic phosphate. A couple of trivia type things regarding ATP. Cells' ATP content is recycled every minute, so this cycle moves quickly. Humans use close to their body weight in ATP daily. That's a lot of ATP. And then obviously if you can't make ATP, you're going to die pretty quickly. So think about that in terms of when we get into genetic disorders and things along those lines, about how that might impact an organism. Getting into enzymes. Enzymes are huge in this chapter. Maybe the whole point of the chapter is enzymes. Essentially, they're going to be catalysts, so they're going to make chemical reactions happen faster. Let's just get into some of the specifics. Here, you guys know reactants and products, but notice that A, B, and C, D are our combinations. What's the word I'm looking for? They're our compounds. Sorry about that. So our reactants have been rearranged. The different molecules, the different elements, if you want to call them elements, have been rearranged. So it probably looks like a double replacement reaction. Maybe you called it a double displacement reaction. Those are our synonyms. So here's our reaction diagram for that reaction that was on the last slide. We've got AB connected, we've got CD connected. There's some sort of a transition state. Then we get our flip, where we got AC and BD. So, excuse me. Notice here we've got this hill we've got to climb. That's called our activation energy, energy of activation. So I teach this to my young students, like picture yourself pushing a rock up the hill. Once you get to the top of the hill, gravity is going to take over and do the rest of the work itself. Same is true here. Once you push these molecules to their transition state, the rest takes care of itself. The rest is a spontaneous reaction. So enzymes are going to lower that activation energy. They're going to essentially take the top off the hill. This picture shows what I'm talking about. So here's our reaction with no enzyme. We certainly can push the reaction up the hill, make it happen, but it's going to happen slower. It's going to take more energy input. When we use the enzyme, we're tweaking molecules. Maybe we're rearranging molecules, but somehow, some way, we're making it so that the hill is shorter and often less steep. So we don't have to go as far. We don't have to input quite as much energy for the reaction to finish. A couple of things that will help you as you move forward. So substrate is the material that the enzyme works on. And then almost all enzymes end in ACE. If you see something that ends in ASE, there's a really good chance that that's an enzyme. The nice thing, again, scientists aren't super creative. An enzyme that works on sucrose is going to be called sucrase. An enzyme that works on lipids is going to be called lipase. So the first part of the word tells you what the substrate is. The second part tells you, hey, this is an enzyme. But, of course, there are some old-school enzymes like pepsin and trypsin that don't follow that naming pattern. We knew about them before we came up with the naming system, so they kind of get grandfathered in. Key part of the enzyme is the active site. That's where the enzyme and the substrate are going to interact. Like it says here on the slide, the structure is designed to fit the substrate, so each substrate has a specific enzyme. And often one substrate might have multiple enzymes for different processes. Here's a picture that shows kind of what I'm talking about. So here's our open enzyme. It's got the active site open and available. Our substrates, in this case, it's going to be a complex of substrates that are going to come together, and they're going to bind with the enzyme. That's going to cause the, notice that the shapes are facing out. The enzyme is going to lower the activation energy. It's going to somehow change the shape of the substrates to make their bonds possible. Notice that we started out with a long and skinny, and now we've got a tall and skinny, I guess. But again, this is going to lower the activation energy. It's going to allow the reaction to happen faster. An example of that sucrase that I talked about, sucrose is a polymer. We've got glucose and fructose connected. Sucrase and water are going to split those into glucose and fructose. So that's just an example of an enzyme at work. Here's the graphical representation of that. We've got our substrate, which is sucrose. So basically this slide combines the last two. We've got sucrose binding to the enzyme. The enzyme's going to change shape. That's going to allow the reaction to happen. That's going to break these two things apart, and we end up with our released substrates. Two types of models. Essentially, these are ways of explaining how enzymes work. There are competing models. There's the lock and the key model, which I called level C. That's probably should be at least a level B. But the lock and key model has kind of been outdated. So the idea is that the substrate fits exactly into the active site like a lock and a key, and that's going to provide a micro environment for the specific reaction. Again, usually it's going to involve some sort of shape change of the substrate, where something might be internal. Think about the protein folding that we talked about in macromolecules. Something might be folded internally on that substrate. The enzyme turns the substrate inside out, and now that key, maybe it's a functional group, maybe it's an aldehyde, something along those lines. That key functional group is external and can now interact with whatever is trying to happen in in the reaction. I'm doing a poor job. I'm trying to generalize and make this as general as possible, and I think I'm losing some of the meaning. But lock and the key model is every enzyme, every reaction has one specific enzyme that's specific to each substrate. Nowadays, we talk more about what's called the Induced Fit Model, and that's that we might have fewer enzymes than we originally thought, because there are often times where we see that the substrate almost fits into the active site, and that almost fitting causes a strain on those chemical bonds that allows the reaction. So I think you can kind of visualize how that would happen. If it almost fits, that means that the enzyme is going to be straining to hold the substrate in, and the substrate's going to be straining to fit into the active site. Kind of like if you're struggling to get into a t-shirt from when you were a little kid, that's going to leave that t-shirt open and vulnerable to tearing and ripping in half, because you almost fit into it. Everything is straining. That's going to allow chemical bonds to break and then reactions to proceed. So some things that impact enzymes, these are each going to get their own slides. So those are the 5 things that we're going to talk about that impact enzymes and enzyme activity. Environment, of course, is going to impact the enzyme effectiveness, especially in if we see elevated temperatures, if we see elevated pHs or decreased pHs, we see massive changes in salt concentrations. All those things are going to affect the functionality of the protein, of the enzyme, which, remember, enzymes are proteins. So essentially what happens in these conditions is the protein unfolds. It's called denaturing. When a protein denatures, like I said, it unfolds. The primary structure tends to stay the same. We tend to be talking about the quaternary structure of the protein. So when that change happens, remember, form controls function. Shape changes, function is going to change. So where that lock and key fit before, or where it almost fit, if we use the induced fit model, where it almost fit, now we change the shape, it won't fit at all. A couple of things that kind of tie in here are cofactors and coenzymes, vocab that we definitely need to commit to memory. And essentially the difference is, are they organic or not? Or are they metals or not? So a cofactor is going to be something like a metal that's going to help an enzyme function better, function faster. And a coenzyme is going to be something like a vitamin that's carbon-based that, again, improves the functionality of an enzyme. We'll leave off here with this video. We'll pick up with enzyme inhibitors in part 3.